Yeah, thanks for coming. Thanks for the invitation and the degree. As I said this morning, I actually didn't go to my defense, or I went to my defense. I didn't go to my graduation, so now I feel like I have a PhD finally, yes. All right, so I want to talk about the origin of eukaryotes, and I'm not going to talk about much data. I'm just going to sort of bring up some ideas that I think are important. And the first one is that this is to me the problem of what we have is something that only happened once, and it happened a long time ago. And these are both really bad problems when you're talking about evolutionary biology. You'd like to see things happening multiple times so you can compare them and see what's important and what's not. And you'd like them to be more recently so that you actually have some evidence of what went on. All right, does that show up? Yes, good. So the other problem with the origin of eukaryotes is there was so many changes. Eukaryotes and bacteria and archaea are also different. And so we have a lot of things to explain. And to me, the simplest way to go through that is to think we need to take all these changes and put them in order. So what happened first, second, third, because certain kinds of changes will precipitate other changes or at least make them more possible. And so if you can figure out sort of what triggers what and sort of put it in a logical order, this makes the problem simpler. However, we usually do that by reconstructing intermediates, and in this case there really aren't any, or there's very few. There's more today, and Eugene will be talking about this very important problem. But for the large part, we don't have too many intermediates. It was, as Stanier said, the greatest single evolutionary change, like leap all at once since the origin of cells. The other thing we like to do is make arguments about plausibility. So something is likely to happen or unlikely to happen, and that mostly comes from looking at modern analogs. So we see this happening a lot in biology, so we assume that in the past it might have been likely. Very simple. Oops. And then the other problem I want to talk about is the age, and this is because old things are hard to interpret. And I love showing archaeology when we do this because we're sort of doing archaeology. And the context is everything. So here's a case that I particularly like where an artifact was found and it was interpreted in the context of the day as this crown. A famous king must have worn this crown. It was buried and, because people see what they want to see. And so modern archaeology goes back and evaluates the exact same artifact and they realize it's actually the top of a bucket. And so it's not we want things to be fancy, we want them to be exciting, and sometimes they're just mundane. Okay, and actually I'm going to take several digressions at this point from the order of events theme, and that's because I think these are actually more important. So these aren't just trivial digressions, these are things that I think are important. And the first is how ecology changes the mode of evolution, or deeply affects the mode of evolution. And I think that discontinuity that Stanier referred to in the origin of eukaryotes is, it's at several levels, and one is at the ecological level. And that's because eukaryotes, unlike bacteria and archaea, eat other cells. And that, to me, this is the most important thing that happened. Because if you think about ecology at the time, it was a bunch of organisms in a population, and they were competing for resources, nutrients, energy, whatever. So they were all in competition for different resources, and the ecosystem was made up of these complex communities. When a cell learned to eat another cell, however, then it's quite different, because now all that population becomes your food and not your competition. So if you imagine the hunter going out in the forest looking for rabbits, he's not competing with the rabbits for food. He hopes there's more rabbits, because the more successful the rabbits are, the more food there is for the hunter. And so they're not, trying to beat each other out for a patch of grass. The hunter wants those rabbits to eat as much grass as possible so that it can eat that. And eukaryotes are the first hunters. Bacteria didn't really do this. They still don't really do this. And so to me, that was actually an earth-shattering event that changed the mode of evolution for these organisms. So they created a niche that didn't exist. Right. And this also trickles down to many levels of how they evolved, because different processes and features will now be selected upon. So let me explain that with this picture. In bacteria, and this is a gross generalization, but bear with me, you have a bunch of organisms that are competing for nutrients and energy, absorption and eating saprotrophically. There might be a nutrient or some form of energy in the environment that you haven't got the capacity to eat, and by evolving the capacity to eat that, you might have an advantage. And this could be a metabolic enzyme that you acquired by horizontal transfer is the sort of famous case, right? And so now you could eat something, in this case the red stars, that you couldn't eat previously, and that's advantageous, and it will be selected for. But this isn't how eukaryotes work. And we don't assume this about many eukaryotes, the ones that we're commonly familiar with. Like, you don't look at the cheetah and say, well, it evolved to run fast by eating things that ran fast, or whatever. It evolved to run fast because of changes to gene expression networks in its developmental biology that changed the morphology of the organism and how it behaved. And my argument is that that's equally true of almost all eukaryotes, even the microbial eukaryotes. So for instance, here's a ciliate. It's going to eat an animal. It succeeds or dies in the environment by its morphology and its behavior. The metabolic network that it's part of is less significant. And these kind of changes are a lot more complex, and they're not simply acquired by getting another enzyme by horizontal transfer, for example. Instead, these are also, like the cheetah, changes to the gene regulatory network that underpins the developmental biology of that single cell. So, in this case, say you have a ciliate, or whatever, this isn't a ciliate, but it's eating some bacteria and its mouth is only so big, so it only eats the small bacteria. Something changes and its cytoskeleton, like in descendants, is a different shape and it can eat bigger bacteria. Now it's got more food availability, and that's selected for. And that is basically a gene regulation expression network that's to do with its developmental biology, the same as the cheetah. So this, to me, again, getting back to the exact same thing, is why this is such a significant change, because it changed the mode of evolution of these organisms. When eukaryotes originated, the way selection worked on them was altered forever, except when it went back, like in the case of fungi, for example, which live much like bacteria do. Okay, the other thing about this that's really important in this debate is, as I said, they basically invented a new niche that nothing else was doing. So it's important to remember that at that stage, competition was limited. There wasn't anything else doing this. So they didn't have to be very good at it. They just had to do something that nothing else was doing. Competition starts up at that point between them. They're still not competing with most of the organisms on the planet. The bacteria, that's their food. They're competing with each other, yes, but they didn't have to be very good at eating other cells. They just had to eat other cells because nothing else was doing it. So a lot of arguments in this field would take the tack that, well, this can't happen because it's so complicated, so how could that just appear? But this is a lot like creationist arguments that, the eyeball is too complex to just evolve. And we all know that the eyeball didn't just, appear. It evolved incrementally. And the first eyeballs might not have been as good as our eyeballs are, but of course that's the case. And it's the same with anything. And it's certainly the same with eukaryotes and their cytoskeleton, their endomembrane system. It started simple, but it was the only game in town, so they were the best at it. And they were not competing with most of the organisms on the planet. They also probably had small population sizes. They could have grown slowly. None of that was very important. Modern predators are also low in population. They do reproduce more slowly. It doesn't actually change the fact that they're very successful at what they do. Now, the second digression that I want to talk about is, how do we think about redundancy in evolution? And we're not very good at thinking about redundancy, I would argue. We tend to think about things in efficiency. So if you have two things that do the same thing in evolution, you assume that one of them is going to disappear. Well, first of all, that sort of parsimonious thinking it's not consistent with the evidence. We see lots and lots of evidence for extremely deep and ancient cases where redundant biochemical pathways, for example, are maintained over long periods of time. They're probably not completely redundant. There's ways in which they're both selected for. And so we can't really just assume that in a case like an endosymbiosis where you double up on things that they just disappear quickly. eukaryotes. We know that doesn't happen. And then this loss of function, when it does happen, when you have redundancy, is treated inconsistently. And this is just a purely logical argument. So for example, in hosts and endosymbionts, we just instinctively, I think, don't treat them the same way. So Eugene and I actually edited a book years ago, and I wrote one of the chapters in the book, and it only had one figure, and it's the most boring figure ever, and this is what I'm going to show here, because I don't think I've ever shown it in a talk. And this is just a logical exercise. When we look at the tree of life, and don't worry about the fact that archaea are monophyletic here, because it's just a logical exercise, and this was before we knew about that. When we see something that's unique in eukaryotes, on the left, we're okay with that. We go, well, yeah, eukaryotes are really different. They just evolved to be different. We don't have any problem with that. When we see something where bacteria are unique, we also don't really have a problem with that, except that we tend to assume that the archaea and the bacteria, or the archaea and the eukaryotes probably changed, and that the bacteria are ancestral. And, that may be true, 50-50. But one of the two changed. That's fine. We don't really have a real problem with the logic there. But when we see something that's unique in the archaea, I would argue, we kind of lose our minds a little bit. We kind of go like, oh my God, how could that be? We start talking about chimeras in eukaryotes. It must be the case. They have a trait they share with bacteria. It doesn't make sense because they're not related to archaea, or they're related to archaea. But in fact, archaea are also just changing. And so it's not necessarily the case that it's as simple as the other two, but it certainly could be. And we're not really very good at puzzling that together, and a lot of chimeric hypotheses come from this logical inconsistency. So getting back to the host, though, and what it was like, the problem with redundancy is because of the mitochondrion primarily, which of course is a big issue in the origin of eukaryotes. Sorry, I keep messing with this thing. So, and the problem there is that we're trying to imagine what was the host like before it had a mitochondrion. And this is problematic because all modern eukaryotes have been evolving for millions and millions and millions of years with a mitochondrion. And the mitochondrion inside that host has changed it a lot. And so trying to work out what the host was like before it had the mitochondrion is not simple. We do take, things like this are the way it's thought about, though. This is just a random metabolic map of the eukaryote that I picked, and the mitochondrions in gray. And so there's some logic that's like, well, if this is what the metabolism of this organism is like, then the pre-mitochondrial metabolism would be like that. And then you look at this metabolism and you go, well, this is terrible. It's inefficient. It doesn't work very well. This organism wouldn't compete. It wouldn't be a very successful organism. How could this have ever happened? Well, of course, it didn't really happen. Well, except that it kind of did, because we have amitochondrial organisms today. Their metabolism isn't like this exactly, but it's actually kind of close. But think about it for a second. It's only that way because they evolved for millions of years with a mitochondrion. The ancestor that took up the mitochondrion didn't evolve for millions of years with a mitochondrion, and its metabolism would have been quite a bit different than this. It would have been much more complex. And in fact, if you look at the organisms that we think are closely related to the ancestor of the eukaryote that took up the mitochondrion, indeed their metabolism is quite a bit more complex than this. For instance, the TCA cycle is extremely common. So there would have been redundancy, and the ancestor that took up mitochondrion was metabolically quite versatile compared to how it's often argued to be. Okay, which brings me back to the order of events. So in this battle of LECA that's sort of happened over the last, 10 years, there's really two camps that emerged. One is about the importance of mitochondria, so the mitochondria early camp, where it's envisioned that the origin of the mitochondria was the trigger that changed everything. And these are largely energetic arguments. Without that energy, without that metabolism, you could not evolve the cellular complexity that's needed to be a eukaryote, for example. That's one version of it. On the other side, the cellular complexity argument is quite a bit older. It goes back to, I would argue, Stanier. And the idea is that the cytoskeleton and the endomembrane system, which to me are the hallmarks of eukaryotes, not the nucleus, it's part of the endomembrane system, but the cytoskeleton and the endomembrane system, the very thing that gives them the behavior that I talked about. So the chasing, the eating, the sensing that eukaryotes are famous and why we love them for come from these two cellular supersystems. And the argument there is that they were required to take up the mitochondrion in the first place because they're required for, say, phagocytosis or the complex trafficking of vesicles and information in the cell that's required for endosymbiosis. But to understand that, we have to understand a little bit about how these organelles, like how those two things go together. How are organelles picked up and how are they integrated into a more complex cell? Now here, again, we're kind of, I really don't like most of the models for this because usually they're really vague. These are just textbook things that I've picked up. And it's one of my most hated things is how they show the membranes all wrong in textbooks. So my students all struggle with this because they've learned a lot of stuff in first year that's just not even true. But even if you get away from the incredibly vague, like magic wand, like, oh, it ate it, it's endosymbiosis, which to me is used like abracadabra, like you don't actually have to explain anything if you just say endosymbiosis. If you get like to the right there, a more complex thing, we see a model like this, where the order of events, and this is a historical thing, is, intuitively people might have said, well, first the organelle was picked up, it was eaten, and instead of digested, it was kept, so it was fixed in the population. Gene transfer followed that, and then protein targeting evolved. And that's when we define something as an organelle, when the protein targeting locks it in, the two cells in together as one cell. That's the most commonly accepted point where you say that an endosymbiont becomes an organelle. And this is, I just picked because it's in science. Well, the problem with this is it doesn't make any sense. It puts the cart before the horse. Because you can't have this order of events, because you have genes existing in the nucleus for millions of years while they're waiting for protein targeting to evolve, and that really can't happen. And so instead, right, and there's evidence that this is false. In fact, the first evidence was from Lilly in a system of a more recent endosymbiosis, a tertiary plastidin and dinoflagellate. I'm sorry this is really complicated, and I'm going to go through it very quickly. But where you can dissect at a very fine scale how the host and the organelle evolved. And you can show that fixation happened many times independently, so the two cells became one very late, and protein targeting evolved before that. And so it's backwards from the model in the textbook, where protein targeting evolves before the organelle. So that's a little counterintuitive. How does protein targeting evolve before the organelle? Well, I'll get to that in a second, but we know it's true because the only other system where this has been looked at the level of detail where you can tell the order of events also says the same thing. And this is in the euglenids, where we have another system where we can dissect the earliest stages of this process. And we can see that protein targeting had happened before the organelle was actually fixed. So protein targeting precedes the organelle, which again makes no sense intuitively. So this is wrong, and I think we should dispense with this as the null hypothesis. There's no evidence that this kind of model is true whatsoever. It might have happened in other situations, but this can't be our null hypothesis. And instead, I would argue there's a different model that actually explains all the data quite a bit better. It's just a model, it's just a hypothesis, but bear with me. So it's a cyclic model, and it focuses on the importance of protein targeting. So you have an organism here, and it's just eating things and digesting them. That's not controversial at all. It happens all the time. So then imagine that it delays the digestion of something for a while. This is also non-controversial. It happens all the time. So let's say you ate something that's photosynthetic, and you just didn't digest it right away because it's still photosynthetic, and that's beneficial. So then what would select for you to maintain that association for longer? To farm it, to keep it in your cells for longer and longer and longer. Selection might favor that, but what would facilitate that? And this is where it gets interesting, because what would facilitate it is protein targeting. So if the host could target proteins to its farmed particles, its food that it's keeping, it could control them and keep them for longer. And I would argue that control takes two forms here. The first is transporters. It could extract nutrients, or it could extract energy or whatever it needed more efficiently if it could target transporters into the membrane surrounding its farmed food. The other thing it could do is control its division. And this means slowing it down. You don't want this thing to divide too fast. You want to contain it so you can, harvest the energy from it, but not have it take over like an infection. So those two kinds of things are going to be host proteins. They're not genes derived from the symbiont. They're your own genes. You're just targeting your own proteins to this nascent organelle in order to control it. So that would be a fairly reasonable thing to expect selection to work on. And then the next step is, having evolved a system to protein target, it opens the gates to gene transfer. So you could get genes from any food particle in your nucleus, and they have some chance of being targeted back to the next food particle. Not the same one, but some future food particle. And if those genes help the cell continue to control and exploit its farm food, they would be beneficial and they would be selected for. So the only way this becomes an organelle is when it spins off and becomes, I wouldn't say permanent, but I guess indefinitely retained. And that could just be, for almost any reason, it just for some reason didn't digest it eventually, because this continues to be a cycle where no matter how long you keep your food, eventually you're digesting it. But if something happened, the lack of signals for phagocytomes or whatever, so you no longer digested it, now you've actually kept the food particle as an organelle. But protein targeting is already evolved, and the nucleus already has a bunch of genes for proteins that are targeted to that organelle on day one. The whole system's already there of day one of the actual fixed organelle. And so you see, this puts things in the same order as the data suggests it is, where protein targeting happens first. And this allows gene transfer to happen and to be retained. And then at the end of it all, you fix an organelle. And it also leads to predictions that fit with the data. Because one of the main predictions is that the host has a lot of genes that are for proteins targeted to that organelle, but they don't come from the organelle. They come from other stuff it ate or whatever. And this actually turns out to be true. If you look at the plastid, it's full of genes that aren't from the same lineage as the plastid. But these are proteins that are targeted into the plastid. Same is true for the mitochondria. A good portion of the mitochondrial proteome is not related to alpha proteobacteria, but came from somewhere else. Originally, these were often interpreted as horizontal transfers, but they could also have been ancestral parts of the cyclic origin of the organelles. And even if you look at modern, really recent things that we now consider to be organelles, bacteria that have been taken up and have protein targeting, these are found in animals, in Pollinella, in Angomonas. They either all, or at least a good portion of the proteins that are encoded in the nucleus and targeted to these bacterial organelle symbionts are not from the same lineage as the symbiont itself. They're picked up from somewhere else. Right. And this is, I can't read that, so I'm just going to skip it. Okay, so getting back to the LECA models, though, the two camps, let's say, they're actually both compatible with this model of how organelles evolve. So I'm not here to say that this one's right and that one's wrong, because I'm not even sure that's the right question. But they're both compatible with this, I would argue, newer view of how organelles are probably integrated into the cell. And instead, I want to ask the question, which you also can't read, which is there even an order of events here to discover? Because I don't actually think it's necessarily the case that there is. I don't think one necessarily happened and triggered the other quite the same way that it's debated. Instead, I want to argue for more of this, which means, as far as I know, it means to wobble forward. And it's, these toys where you pull one string and then the other, and the monkey works its way up to the top? Are you familiar with these things? We had them when we were kids, at least, but I don't know. Maybe it's an app now. Anyway, what I want to argue is that there's selection to pick up, digest, and keep these farmed cells. But there's also selection, and at the same time, the selection for the complexification of the cell that's doing this are actually compatible. And instead of viewing these two sort of things as opposites, and that we're trying to figure out which one might have precipitated the other, I would argue that they would actually facilitate each other. And so a little bit of cytoskeleton in an endomembrane system might have precipitated the pickup of the first farmed food, say, but that itself would then lead to selection for more complexity, say, in the endomembrane and the cytoskeletal system, which would translate back to more likely to pick up more cells, more likely to do farm food. And so the origin of the mitochondria and the complexification of the endomembrane system and the cytoskeleton, I sort of see as two things that could support each other. They could push each other. They could sort of wobble forward, as I was saying. They are the impetus for each other to both evolve at the same time. And so I would like to see more wobbling forward. I think that might actually be a way to unite these two things and understand them a little bit better. I have no idea if I'm over time, so I'll just say thanks and thank the Moore Foundation and my host.
Okay, good afternoon. The first and main thing is actually to congratulate Patrick with his honorary degree. Sorry, with his PhD, because he didn't really have. And also thank Patrick for the excellent, brilliant, provocative, at the same time easygoing talk. It's not an easy act to follow, but we should try in a different key. And of course, to thank wholeheartedly you for organizing it all, making it all possible, inviting us, and entertaining us here so beautifully. Okay. So we're going to discuss for the next half an hour the origin of eukaryotic cell, and essentially do that in a far more technical way than Patrick did. Trying to understand, to speculate maybe a little less, and to understand where do eukaryotic genes come from, and what that can tell us eventually about the origin of the eukaryotic cell. But of course, we need to start with models. And the models are argue three kinds. The first kind, like in A here, sort of direct ortho-evolution of prokaryotes into eukaryotes. Completely unrealistic. What is shown as B here is more or less mitochondrial late evolution of something that Patrick has touched on. More or less evolution of complexity before the acquisition of mitochondria and the acquisition of alpha proteobacterium that became mitochondrion as sort of the final step of eukaryogenesis. And finally in C, what you see here is mitochondria early, whereby the acquisition of the alpha proteobacterium and the symbiont triggered and drove eukaryogenesis. What can we, I should note maybe that the acquisition of alpha proteobacterial symbiote that became mitochondrion is not a concept and not a hypothesis. It's a plain fact. What we have to figure out is how this happened, when this happened, and how this either triggered or followed essentially the process of cellular complexification that led to eukaryogenesis. So, eukaryogenesis. LECA, last eukaryotic common ancestor, already was quite a complex cell that had all these important intracellular organization, which I agree with Patrick completely. The cytoskeleton and the endomembrane system are the real hallmarks. The nucleus is just sort of the derivative of the endomembrane system, which of course is functionally very important too, and it had the double flagella apparently. Anyhow, it was a fully fledged eukaryotic cell. So what we have to figure out, how all this complexity came to be. How essentially life produced all that complexity, followed by all that enormous diversity of eukaryotes, some of which Patrick beautifully showed, starting with the relatively simple but we shall see that relatively simple prokaryotic cell. So, there are many models. Indeed, Patrick sort of was dismissive of them. And maybe for a good reason they're speculative. But we have to start somewhere. We have to, if you will try to falsify certain models, and for that you need them. So several possibilities. One, the most simplistic, one symbiosis. Asgard archaeal host. Don't worry if you don't remember what Asgard is. I think you do, all of them, but if you don't, I will introduce them. Asgard archaeal host, alpha protobacterial symbiont, everything very simple. Second possibility, multiple symbiosis, or actually I think Patrick was, part of Patrick's talk was sympathetic with that idea, also Asgard archaeal host. And the third possibility, something a bit more exotic, but as we shall see, not implausible. More two symbiosis, first bacterial host that accommodates the archaeal cell, then later alpha protobacterial symbiont coming and becoming mitochondria. So, enter Asgard. Just in case Asgard is the Norse pantheon of Nordic deities, and they are supposed to inhabit this beautiful city. So, everything changed a decade ago. It's actually a bit of a celebration this year. I have already spoken at a couple of celebratory symposia. Let's celebrate again. This is basically a discovery that Thijs Atema and his colleagues, then at Uppsala, and hence the Nordic deities, made by painstakingly putting together genomes of novel variety of archaea from metagenomic data. At that time, and even essentially even now, it was practically impossible to grow these things. At that time, no one absolutely had any idea what to do about them, but Thijs and his colleagues painstakingly put together these genomes, and lo and behold, what happened? What happened was that these newly discovered archaea were the clear sister group of eukaryotes. Until this discovery, there has been a lot of debate. Where do the eukaryotic, let's say at this moment, eukaryotic ribosomal RNA and other universal genes comprising the information transmission systems in eukaryotes, where do they come from? Outside archaea? Is it the sister group of all archaea, or somewhere from the inside? It was a difficult debate hinging on technicalities of phylogenetic analysis. As it often happens, it's the new data that solved the problem. Even simple phylogenetic analysis put them here. So eukaryotes come from within, and the sister group is this novel variety known as Asgard. This seminal work stimulated a lot of developments, expansion of Asgard diversity, and as the Asgard diversity was expanding, many new clades of Asgard archaea have been discovered, and the eukaryotic branch was going deeper and deeper into the Asgard part of the archaeal tree. This is our little contribution collaboration with Chinese colleagues, where we significantly expanded the Asgard diversity once again and tried to carefully study the phylogeny and came up with similar conclusion that eukaryotes looked like the sister group of so-called Heimdall archaeota, named after one of those Nordic deities. And here we introduce a Chinese deity for a change. The remarkable feature of Asgard archaea feature to remember is that these archaea, much more than any other prokaryotes, encode homologs of the so-called ESPs, eukaryotic signature protein. Those ESPs show sort of patchy, mosaic distribution across the diversity of Asgard archaea, but at least some of them are particularly enriched in the Heimdall branch. Interestingly enough, these are essentially domains of these eukaryotic signature proteins that you find in Asgard. And many domain architectures of Asgard proteins are actually not eukaryotic, indicating that if these were indeed inherited by eukaryotes, a lot of change and rearrangement happened along the way. Further extension of Asgard archaeota, pushing eukaryotes up and up and up. Now, at this time, this is I think the best we have for the moment, and eukaryotes come up as a sister group of so-called Hodarchaeales order. I strongly suspect this is not the end. Something even closer to eukaryotes still will be discovered. So, this is how the eukaryotic connection of Asgard archaea looks for the moment. And one more thing, important thing to notice about Asgard archaea is, which sort of makes their role in eukaryogenesis particularly plausible, is that they exist as consortia with various bacterial cells, most notably the proteobacteria. The next digression that I want to do is a reconstruction of the virome of the LECA. Since some of you may have noticed that we do a lot of work on virus evolution, that's my favorite subject. So we try to reconstruct ancestral viromes. When we do so, mapping different groups of viruses on the branches of the eukaryotic evolutionary tree, and doing a evolutionary reconstruction using the parsimony principle, basically. And what we come up is a very complex, diversified virome of the LECA. This is not a surprise. The surprise comes here when we start to investigate the roots, the origins of these components of the ancient eukaryotic virome, of the virome of the LECA. What we see is that all major groups of eukaryotic viruses have actually bacterial, not archaeal, as one could perhaps intuitively think, origin. And interestingly, Asgard archaea, as we and others also investigated, are host to typical archaeal viruses, no trace of the eukaryotic virome ancestry. Why is that? I don't know, of course, but the main idea that comes up here is that this is because of what I call the great membrane divide. The membranes of archaeo and bacterial cells are dramatically different. They have different lipid composition and different chirality of the membrane phospholipids. That's shown here. And eukaryotes have inherited, most interestingly and rather mysteriously, the bacterial the typical bacterial membrane structure. This is the case both for the plasma membrane of eukaryotic cells and the endomembrane system, including the nuclear membrane. So that is the idea then. That is the hypothesis, that the archaeoviruses were excluded from emerging eukaryotic cells because the entrance and egress were hindered by bacterial cell membrane. And this, I think, provides support for models of eukaryogenesis that postulate continuity of ancestral bacterial membranes. Remember in the beginning, I show these more complex models that include first a bacterial host, potentially delta proteobacterial, as proposed in this so-called syntropy hypothesis by Puri, Lopez Garcia, and David Moreira, which possibly engulfed an Asgard archaeal cell, and this was followed by the acquisition of the alpha proteobacterial endosymbiont, keeping the integrity of the bacterial membrane all the time and explaining the composition of the LECA viral. I think it is a beautiful model. The whole problem is that, as I will show in the second part of my talk, which will be dedicated to specific phylogenomic analysis, the whole problem is that it's probably wrong. So to the second part, delineating contributions of different archaeo and bacterial phyla to eukaryogenesis by doing comprehensive phylogenomic analysis, work done primarily by Victor Tobiasson, who until last week was a postdoc in Canada. So we'll quickly go through the methodology. Databases, large databases representing eukaryotic diversity. We are looking at proteomes, of course, here, and a dedicated database of prokaryotic genomes, specifically enriched by Asgard archaea, because these genomes are not necessarily complete, and so they needed to be added. Importantly, what we did here was requiring that all prokaryotes except for Asgards were represented by conserved genes, by genes present in at least about 2/3 species per or genera per lineage. We shall see that this is important to rule out late horizontal gene transfer. So then they, of course, as always in such studies, develop a bioinformatic pipeline to analyze the evolution of all genes that are shared by eukaryotes and prokaryotes, perform a fairly sensitive sequence-based search, sequence profile-based search, in order to identify as many orthologs between eukaryotes and prokaryotes, both archaea and bacteria, as possible. And then use these sets of orthologous genes to make phylogenetic trees, and analyze these phylogenetic trees using so-called constrained tree approaches, which means that we try to falsify different phylogenetic hypotheses and force certain topologies of phylogenetic trees, for instance, clustering of Asgard archaeal proteins with eukaryotic orthologs in a phylogenetic tree, and assessing the likelihood of each of these models, each of these alternative phylogenetic hypotheses, using so-called expected likelihood weights developed by old but important work by N.T. Rambo. Essentially, we cannot go into the details of this, but essentially this is analogous to assessing the confidence of the inferred ancestry. And for functional notation, we use the excellent KEGG database. And preempting myself, what we'll see here is domination of Asgard archaea association across a broad variety of cellular function. So here is maybe the key result of this analysis. What we see here, these light blue rectangles, are the contributions of Asgard, the contributions of Asgard archaea inferred from phylogenetic analysis, and all this colorful stuff is the bacterial contribution. You can visibly see the domination of Asgard archaeal origin across both metabolic functions and information. Central dogma here, information transmission functions, where the dominance is particularly dramatic. This is not, when it comes to information transmission, to translation, transcription, replication, this is not unexpected. Even though the degree of the dominance may be so, but we more or less know that these things are of archaeal origin. The breadth of Asgard association across cellular functions is actually quite unexpected. This was not observed previously, and I think there are two reasons why this was not observed previously. One is that we simply had the increase greater than previously tried diversity of Asgard archaea. The second one, which is an important methodological issue, is this pangenome definition, whereby we exclude genes that are found with narrow phylogenetic distribution in bacteria and accordingly exclude late horizontal gene transfer. As you can see, it's not even so important what definition of the pangenome is adopted. It's important to exclude the most narrowly distributed group, in all likelihood coming from later horizontal gene transfers. If you do so, you clearly observe that domination of apparent Asgard origin of the eukaryotic genes. And they have, well, just show you more specifically various functional group of eukaryotic genes, and in essentially all of them, we see the same pattern. Different degrees of this Asgard domination, but the overall pattern is just the same throughout almost all cellular functionalities, with some, again, even more detail on this, with some notable exceptions. We evidently are very interested in alpha protobacterial contributions, and those are detected all right, but these are mostly limited to specific mitochondrial functions such as oxidative phosphorylation, synthesis of iron sulfur clusters, glutathione synthesis, and a few others, functions that are highly specific to mitochondria. You can even some of those functions actually seem to have chimeric origin, with some contributions coming from alpha protobacteria and some from Asgard archaea. For all other bacterial phyla, we see the same pattern, namely a diversity of small contributions to apparent contributions to eukaryogenesis without dominating functional themes. It's particularly interesting, this Myxococcota, because these are the bacteria that form syntrophic associations with Asgard more than others. There is some specific functions attributable to Myxococcota, but really not very much. Interestingly, as an illustration, if we take, for different bacterial phyla, if we take those pathways where the contribution is most prominent, still, in practically all cases, as you see from these pale blue columns here, the Asgard archaeal contribution is greater than the contribution of the respective bacterial phyla, again, emphasizing the dominant role of, apparent dominant role of Asgard genes in eukaryogenesis. And the final thing that I want to do here is to show you some results of the analysis of stem length in phylogenetic trees. This is a famous paper by Tony Gabaldon and Alexander Spitis, which happened to come out to have been published more or less simultaneously with the discovery of Asgard archaea, and accordingly did not include data, I've got branches in the tree. What they claim here are shorter branches for alpha proteobacteria than for archaea or other bacteria, suggesting late acquisition of the mitochondria. Well, we repeated that analysis as best we can. What we actually see, remember now we have the Asgard archaeo data. What we see are longer stems for genes of bacterial origin compared to Asgard. The conclusion from this is that probably this is not a particularly great type of data to infer timing of the acquisition of the symbionts or anyhow timing of different events, because in all likelihood this is due to the accelerated evolution subsequent to horizontal gene transfer. Similar thing for different cellular functions. So what does it tell us about the likely scenario of eukaryogenesis? Actually, a rather straightforward scheme now seems to me the most likely one, informed by this comprehensive phylogenomics analysis, whereby cellular complexity, as actually suggested also in Patrick's, at least started to evolve within the Asgard archaeal cells, supported by all these eukaryotic signature proteins, as well as by continuous influx of bacterial genes with diverse functions, with the alpha proteobacterial and the symbiosis coming relatively late, resulting in limited addition of genes involved in specific, primarily energetic mitochondrial function. As Patrick mentioned, more than a decade ago, with a very good time in anticipation of the discovery of Asgard archaea, we edited this book. And we also had a chapter there describing what we call dispersed archaeal eukaryote. Dispersed because homologs of eukaryotic proteins showed patchy mosaic distributions in archaea. And this was more or less the model that we proposed there, which is, if you may notice, very similar to what we are proposing now, except we did not know about Asgard at the time and had to sort of postulate the evolution of the more complex archaea. So, ahead to the past, so to speak. This is still unpublished data, but you can read it in the preprint. And so such are my conclusions from comprehensive phylogenomic analysis as of today. Overall, Asgard dominance in the ancestry of eukaryotic genes across the entire diversity of cellular, metabolic, other information transmission, and other functions. So best compatible with piecemeal bacterial contribution to evolving Asgard archaean, or followed by relatively late alpha proteobacterial endosymbiosis. Of course, we need to exercise a lot of caution, considering especially that some important groups of bacteria, such as the Myxococota, which I mentioned, are sampled poorly. The conclusions may change. For the moment, as much as I love those models with the continuity of the membrane and two consecutive symbioses, the data do not seem to support this as we see as of now. Okay, so the people who contributed and a variety of people who contributed to previous work that I mentioned, and some essential discussions with colleagues. Thank you very much. Happy to answer questions, probably after Patrick, I don't know.